Okay, so today we're going to review some general principles of pharmacology, and we're going to go essentially through the LADME system, and we're going to review each of these concepts individually. So of course, in order to understand some of the basics of pharmacology, you should go back to some of the biochemistry concepts that you learned hopefully in undergrad, but as well as our own biochemistry section. And those topics are listed here. These are the kind of vocabulary words, so to speak, that you need to understand in order to successfully understand the science of pharmacology. And so what you should be able to do by the end of this lecture is basically define each of these words. And it's going to be important for you to do so because all of the descriptions of some of these drugs that we're going to be learning about for the whole year are going to be described by these terms, and so it's really imperative that you understand what these terms mean and how you relate these terms to clinical decision making. Likewise, here's the learning objectives. I'm not going to read them to you. I'm sure you can read those by yourself. So just as a brief introduction, pharmacology is obviously the study of drugs or medicine. Modern pharmacology, as we know it, really kind of began in the 1880s in Europe. And just as kind of a quick who's who, the medicinal chemists who work for big pharma companies physically make the compounds. Those compounds are then sent to pharmacologists like myself, who will do preclinical evaluations. And so what we're going to do is we'll look at the way the compound works, typically in vitro first, followed by in vivo second. So we'll start with maybe something like cell lines, and then we'll progress that compound. If it looks promising, we'll put it in animal models of the specific disease we're studying. And eventually, if the drug is good, it gets approved by the FDA, and physicians like you will one day prescribe the drug. Pharmacists will dispense the drug, and the FDA will monitor the drug in the entire population and report any adverse effects or toxic effects, so that both physicians and scientists are aware of what these drugs do in vivo. So the first concept in terms of pharmacokinetics, and that's really kind of the study of how these drugs interact with the system and what dosages and concentrations should be reached in order to produce a physiologic response. So the first concept is called LADME, and so the first concept is called liberation. And so LADME stands for liberation, absorption, distribution, metabolism, and excretion. And so essentially what you're doing is you're going from pill to different portions of the body, distributing throughout the body, where it gets metabolized, and then finally it leaves the body after the physiologic response to the physiologic effect has occurred. And so it's kind of like the big picture. Again, the drug goes in, it gets physically dissolved, so the capsule or the tablet gets liberated. The compound gets liberated from that capsule or tablet, and it gets absorbed. And we can model this mathematically by looking at the body in terms of different compartments. So for simplistic sake, we'll just say there's a central compartment, and the drug exists between the concentration of free drug versus bound drug. And that kind of determines the biological activity. Some drugs are not biologically active when they're first administered. They need to be biotransformed, which is a fancy word of saying metabolism. And then this drug becomes active in this compartment, and then there exist reservoirs where some of this tissue can exist between these bound and free states. And the drug, depending on the concentration, can move in and out of these reservoirs. When the drug reaches the site of activity, typically it's governed by the expression of a specific receptor. We typically call that therapy, or we associate that effect with therapy. When drugs bind to unwanted receptors or in anatomic locations where we don't want them, that's usually the beginning of a side effect or a toxicity. And of course, at some point, depending on, again, the chemistry of the drug and how tightly some of these drugs are bound to these receptors, the drug at some point will be excreted. And then the way we kind of measure that is by mathematically modeling clearance. And clearance is primarily governed between both hepatic and renal elimination, but there are other modes of clearance. So this is kind of a brief overview of the whole system, we're looking at. And that leads us into the second phase of the LADME component, which is absorption. Okay, and so at this point now, the drug has been liberated from its capsule, and now it's going to be absorbed. These are different processes by which a drug can be absorbed. It can be both passive and active, and basically the different physiologic outcomes are governed really by the chemistry of the drug. So some drugs are more lipophilic, some drugs are more hydrophilic, some drugs are large, some are small. And so all of that determines how the drug is going to get in, if it's going to get in. Earlier we had mentioned that ionization plays a big role in terms of the availability of a drug. And that concept of availability will be further expanded upon later in this lecture. But essentially here you've got the pill becoming liberated, and essentially the drug, at least in the gastric component, so this is an oral tablet, right? In the gastric component, or the gastric portion of the system, the drug exists in a continuum between its inactive, its salt form, and its active, its ionized form. Okay, and so obviously at low pHs, this particular weak acid is going to exist primarily in its inactive form. Once this drug gets absorbed through the lipid and mucosal barrier, so we're leaving the stomach and we're entering the blood plasma, there's a dramatic pH change. It's going from about 1.5 to 7.5. And this process is the process of absorption. And notice here that now this weak acid is going to dissociate in a much more dramatic pace. And now the ionized form of the drug, as designated by the A-, exists in 1,000 to 1 ratio. And so this is how a lot of these drugs get absorbed. Many drugs are weak acids. They get into the stomach, and once they cross that lipid mucosal barrier and they experience this change in pH, this weak acid dissociates, and this reaction going forward is favored because of the energy, and you get a large accumulation of this active drug that is a ionized form of the chemical. And that's essentially how all of these drugs at some point become active. Now we can slow that process down by changing the pH of the stomach, or by encapsulating the pill with a coating. Sometimes you see enteric-coated aspirin and enteric-coated other drugs for sale in the market. These just have kind of a protective chemical surrounding them so that the process of absorption gets delayed. And we'll go through clinical examples where you would want to have delayed absorption so that the drug sticks around for longer periods of time, essentially. Other components of drug absorption is that essentially the process of uptake from the site of administration can be extravascular or intravascular. And so the route of administration really is important in terms of how efficient and how quickly the drug can be absorbed. And so we'll talk about these routes of administration in the next lecture.